GGE Biplot. This is a powerful graphical method used to compare genotypes, study stability, and evaluate environments. Instead of interpreting long tables of data, GGE Biplot converts complex results into a simple visual graph using principal component analysis. It helps answer important questions such as: which genotype performs best? Which genotype is most stable? Which environment is ideal for testing? And which genotype wins in specific locations? The GGE Biplot are of different types like mean versus stability, which one where polygon view, genotype ranking, and environment comparison. Now let's interpret the GGE Type 1 Biplot for grain yield with an example from RAISINS. First, what is a Type 1 GGE Biplot? This biplot is constructed using genotype effects and genotype by environment interaction after removing the environmental main effect. How reliable is this biplot? The first two principal components explain 64.33% of the total genotype plus genotype by environment variation, with PC1 contributing 41.67% and PC2 contributing 22.66%. This indicates that the biplot provides a reliable summary of the data. So, what do PC1 and PC2 represent here? In this Type 1 biplot, PC1 and PC2 represent the two major interaction patterns extracted from the GGE metrics. And what does this plot mainly help us visualize? It helps us understand relationships among environments, genotype interaction patterns, and adaptability of genotypes to environments. Now, how do we read the plot? The blue vectors represent environments, while the red points represent genotypes. Which genotypes are considered stable? Genotypes closer to the origin are more stable, meaning their performance is consistent across environments. Which genotypes are adapted to specific environments? Genotypes positioned in the same direction as an environment vector tend to perform relatively better in that environment. For example, genotypes located near the direction of E7 and E10 are more adapted to those environments. Now, let's look at the environments. How do we identify environments that effectively differentiate genotypes? The length of each environment vector indicates its ability to differentiate among genotypes. Longer vectors indicate environments that are more effective in differentiating genotypes, while shorter vectors indicate less discriminating environments. Environments E13, E9, E5, E1 and E10 are the most discriminating because they are farther from the origin. What does the angle between environment vectors indicate? Acute angles indicate positive correlation, obtuse angle indicate negative correlation, and right angles suggest little or no correlation. Overall, this Type 1 biplot is mainly useful for understanding interaction structure and environmental relationships. Now, performing this analysis becomes easy with RAISINS, your all-in-one platform for complete statistical analysis. In RAISINS, simply upload your dataset in CSV or Excel format. Choose environment, genotype, and response variables, then click Run Analysis. The platform instantly generates complete results along with all 10 types of GGE Biplots with interpretations. You can customize graphs, compare genotypes, analyze environments, and directly download publication-ready figures. RAISINS, the future of research, begins here.